नमस्कार दोस्तों, welcome to channel Toxicology on board. In this video, we'll be discussing about DABT topic micronucleus assay. So let's directly dive into the question. So the question is a treatment related increase in number of micronucleated cells in a genotoxic study indicates that what type of adverse effects. So we need to understand treatment related increase in number of micronuclear cells in genotoxic study. Okay. So option A is chromosomal translocation. Option B is toxicity to bone marrow cells. Option C is DNA point mutations. And option D is clastogenicity and aneuploidy. So the right answer is in green, that is uh, clastogenicity and aneuploidy. So let's see what is the explanation and the reference which is given by American Board of Toxicology. Okay, the micronucleus assay measures the amount, measures the number or chromatic erythrocytes which are remnants of the clastogenic DNA damaged in mature red blood cells. So already we know that RBC, erythrocytes know they are enucleated cells. So there is a remnant of this clastogenic DNA in this matured RBC. By detecting these remnants, we will be understanding the clastogenicity and micronucleus assay. The assay is micronucleus tester. Okay. So the technique also detects, apart from clastogenicity, it is also used, this micronucleus assay is also used to understand aneuploidy. So if you want to learn in detail, please go back and refer cassette and 8th edition, page number 466 and 467. Okay, so let's understand from this question in detail about in some of the details about micronucleus assay. The micronucleus assay is a toxicological and cytogenetic tester that detects chemical substances or agents that cause chromosome damage. Okay, if you look into the structure of our DNA, chromosomes will be there. So these are the chromosomes, both in vivo within living organisms and in vitro, that also in cell cultures. Okay, this micronucleus assay we can understand it is understand the chromosomal damage. Okay, so in that also what type of damage means clastogenicity as well as aneogenicity. Okay, this micronucleus assay works by identifying small membrane bound bodies called micronuclei. So these are the micronuclei within the cytoplasm of a cell. These micronuclei are formed from either fragments of chromosomes. Okay, these chromosomes are then the fragments of chromosomes that break, that is clastogenesis, or whole chromosomes that fail to migrate properly during the cell division, that is aneogenesis. Okay, so aneogenesis is complete chromosomes, whole chromosomes. Okay, they have to go to the daughter nuclei, but they fail to migrate properly during the cell division. Okay, the frequency of this micronuclei in treated cells or organisms serve as an indicator. So there is a chemical you are studying on certain test species, test system, either in the cells or in the body of that particular organism. Okay, then whatever this chemical, it is causing damage, it will serve as an indicator of this genotoxicity with higher frequencies suggesting greater chromosomal damage. So this is a cell anogenic treatment, clastogenic treatment. In case of anogenic treatment, whole chromosome loss is there. So there is a formation of two daughter nuclei. Here there is no there is a loss in chromosome. Whereas in this case, complete, you can see that micronuclear formation is there. In the case of clastogenic treatment, what is happening? Acentric fragment loss. Okay, so this is here the part of the chromosome. Here, this chromosome, complete loss of chromosome is taking place in the case of androgenic treatment. See, probably I might not be able to explain you in better words. I would request you can pause the video, go back and understand this type of damage. What is anogenicity? What is clastogenicity? This definition should be there in your mind to understand this micronucleus assay. Okay. So how this micronucleus assay works is there are four steps: the cell treatment, cell division, cytokinesis block, which is optional, then micronuclear formation, and fifth one is scoring. So understand what is cell treatment. Cells or organisms are exposed to a test substance. Here the test substance is nothing but the chemical which is causing you the DNA damage, such as it might be either chemical or it might be a radiation. Radiation also causes DNA damage, right? Second one is cell division. Cells that are dividing are particularly important for the assay. So the cells in which you are doing the study, the effect of your chemical, they should be under cell division state. Okay. Cytokinesis block. In many protocols, study protocols, a chemical like cytochalasin B, okay, it is added to block the cell final division. Cytokinesis stage को arrest करने के लिए you will be adding a certain chemical called the cytochalasin B. This results in binucleated cells. So two nuclei will be formed with the cells with the two nuclei, ensuring that micronuclei can be definitely linked to a single cell division. Right? Next, micronuclei formation. During cell division, damaged chromosomes or acentric fragments are left behind and form as micronuclei in the cytoplasm. Scoring. The cells are then stained, okay, and examined under a microscope to count the number of micronuclei, right? So, what are the type of agents which are detected? As we have seen, the question is also the same. In our option number D was to understand what is clastogenicity and anugenics. So, this micronuclear assay can detect agents that cause damage through two main mechanisms. One is clastogenic agents, second one is anugenic agents. Clastogenic agents induce breaks in chromosomes. Chromosome will be there, right? There is a break in the chromosome, creating acentric fragments that become micronuclei. Small micronuclei will be there, right? Then anugenic agents disrupt the mitotic spindle. If you look at a cell, no, uh, in the middle layer, you will be having the spindle fibers that will be pulling the chromosomes in poles apart in opposite direction. So disrupting this mitotic spindle, because of which the chromosome will remain at the center of that cell division stage only. Okay, leading to a whole chromosome being excluded from the main nucleus and forming micronuclei. Okay, here the micronuclei in the cluster, it will be a small chip part of the chromosome. But in the case of aneurysm, whole chromosome is left behind because of the disruption in the mitotic spindle fiber. Okay. What is the application of this micronucleus? The micronucleus assay is a versatile and widely used tool for number one, genotoxic testing, second for biomonitoring, third is for radiation biodosimetry. So let us understand what is genotoxic testing. Micronucleus assay assesses the potential of new chemicals. Okay, those chemicals which are in the drug discovery stage, or the certain chemical which is at a very for which there is no toxicology data is not available. So potential of the new chemicals, pharmaceuticals and cosmetics to damage DNA. If you look at the drug discovery cycle is there, no, for example, any chemical which we want to we want to develop it as a pesticide or agrochemical, you have to do some preliminary screening, then you will be doing some genotoxic test. In that AIMS assay, you will be doing battery of three DNA damage test for the preliminary. First, you will be doing AIMS test. Okay. Eventually, we will be doing micronucleus test, and you will be doing chromosome aberration assay. So, in that micronucleus assay is also very important. The preliminary screening of any chemical, that chemical which has a potential to develop as an agrochemical or pesticide, or the chemical which can be developed as a pharmaceutical, where the end user will be either human or the veterinary animal, and cosmetics. So as we know that there is a almost complete ban on animal testing on cosmetics. So if you want to use a new ingredient which could be a part of a cosmetic formulation, then you have to do some preliminary genotoxic test. In that also, this particular assay, micronucleic assay, play a key role, right? If we know that certain chemical is causing the DNA damage, that chemical will be failed at the early stages only of the developmental cycle of that particular chemical or drug, you can say. Next one is biomonitoring, evaluating genetic damage in populations exposed to genotoxic agents in the environment or workplace, right? Particularly biomonitoring is done in industrial area where the workers are working. There you will be taking their samples, epidemiological studies also, right? You will be taking either urine samples or plasma or serum samples. There we will be checking if there is a continuous frequent exposure, repeated exposure to a certain chemical, whether it is causing any DNA damage. Similarly, environmental also. Environmental risk assessment, impact assessment reports are there, ecological testing will be there, epidemiological surveys will be there, where biomonitoring is very, very important. In certain region, because of a chemical spill or in the water body, certain chemical concentration has been increased in the groundwater, and the people who are consuming that groundwater, if it is causing any DNA damage in that particular cohort of population, that for such kind of studies, biomonitoring is important. Third one is radiation biodosimetry, reconstructing the dose of ionizing radiation. So first two were more like applicable to chemical. The third one is for radiological substances like uranium is there, plutonium is there, right? These are the agents which are the ones which are causing DNA damage because of the property of this radiation property. So reconstructing the dose of this ionizing radiations absorbed by individuals, especially after accidental exposure in a certain lab or experimental area or excavation area, mining area, if a certain group of workers or populations or scientists are exposed to certain radioactive material, if it has caused certain type of damage, by studying these type of studies, we can lower the doses. Okay, the scientists who are handling such radioactive materials, which have potential application in the case of cancer treatment or like that, by using these kind of assays, you can monitor the dose, you can decide the doses, you can do the reconstructing the dose of this ionizing radiation. In that case also this micronucleus will assay will help. So hope from this video you have learned about what is micronucleus assay, what type of main important thing you have to do, type of genotoxic assay, AIMS assay, what type of DNA damage it is detecting, micronucleus assay, what type of DNA damage it is detecting, chromosome aberration, what it is doing. So type of assay and type of damage it is used to detect. Okay. In my previous videos, I explained in detail about genotoxic test, why we are doing a battery of tests, because from a single assay you cannot identify all type of damage. For that reason, you have to use a battery of tests. Within names also, you will be having different strains, TA, 96, 95, 10, some 1000 something numbers will be there. So similarly, you will be doing in vitro genotoxic test, in vivo you will be doing, okay, human cell also you will be doing, lymphoma is there, T-cell lymphoma assay is there. So what type of assay and what type of damage it is used to identify, that is very, very important. One question on genotoxicity you will get in the exam, that is for sure, based on the previous papers or like that. So in my videos now I have given the list of the tables also from Clayson. You can see there also. Okay. So thank you so much. Be curious. For any questions, please you can write down a mail to this particular mail ID, and like, subscribe, and follow my YouTube channel Toxicology on Board. All the best for your exam.